Okay, so welcome to this next video in which we are discussing the use of glucocorticoids in the treatment of asthma. Okay, right, so we're currently discussing the pathology of asthma. So we're discussing the primary exposure to the allergen and how that triggers a humoral adaptive immune response. So we've seen that the professional antigen-presenting cell will phagocytose the allergen and then chop up that allergen and put fragments of the allergen on its surface bound to the major histocompatibility complex class 2. Okay, these professional antigen-presenting cells will then go through the lymph vessels to lymph nodes, and they'll present this to CD4 positive naive T cells. Okay, now when they find a CD4 positive naive T cell that has a T cell receptor which is capable of binding to this allergen fragment that the antigen presenting cell is presenting, then it will activate that CD4 positive naive T cell and that will give it signal 1. It will then also need to express co-stimulatory molecules on its surface to give the CD4 positive naive T cell signal 2. That CD4 positive naive T cell will then go through this autocrine signaling process where it produces interleukin 2 and also the interleukin 2 receptor alpha component, and this then leads to signal 3. That CD4 positive naive T cell then differentiates into a T helper naught cell, divides within the lymph node to give a whole population of T helper naught cells, and those are then going to differentiate into T helper 2 cells due to the actions of interleukin 4. Okay, these T helper 2 cells are then going to help with the B cell activation process. So allergens will also come in the lymph vessels to the lymph nodes and will bind to B cell receptors which have an affinity for binding to that allergen. Okay, so you've got a huge great population of B cells within your B cell follicles. These are currently naive B cells. They've never seen allergen before or antigen before. Okay, now some of them will have B cell receptors which just so happen to bind to this allergen, okay, and remember we're not totally sure which portion of this a humoral adaptive immune response is different between the allergic individuals and the non-allergic individuals. So what I'm describing is what must happen in the allergic individuals, but we're not sure which bit of this goes wrong in the non-allergic individuals, or right, if you like. Okay, so what will happen then is the allergen will bind to a B cell receptor on the surface of a B cell, okay, and that will be internalized. The B cell will then present allergen fragments on MHC class 2. It will have co-stimulatory molecules on its surface, and it will find a T helper 2 cell also within the lymph node. And generally to do this, what happens is these activated, these activated B cells here, okay, that have had the antigen bind to their B cell receptor, what happens is they migrate to the edges of the B cell follicles. So if I get my picture of the lymph node again, so the T helper 2 cells are going to be in this sort of blue region here, in the green region, okay, so what's going to happen is these B cells that have their allergen fragment on MHC class 2 and also co-stimulatory molecules on their surface, they migrate to the edge of the B cell follicles to meet the T helper 2 cells, and then what can happen is you can form an immune synapse between the B cells and the T helper 2 cells. Okay, this then causes the activation of the B cell. So the T helper 2 cell gives the B cell T help, basically, what's known as T cell help. Okay, and this is firstly through the activation of all of these proteins that are involved in the immune synapse, but also the T helper 2 cells will secrete certain molecules which will stimulate the B cell. Okay, now what will happen is the B cell will divide and divide and divide and produce a whole population of B cells that is based on it, and this produces a germinal center within the B cell follicle. Okay, and these B cells that you're producing, these are now what are known as germinal center B cells, okay? So they're slightly different. They look slightly different to the naive B cells. So these are germinal center B cells, okay? Often abbreviated to GC B cells. GC B cells. Okay, now these do look slightly different from the naive B cells. The naive B cells, which are sitting in the B cell follicles, they have absolutely tiny amounts of cytoplasm. Okay, so if we draw a naive B cell, it will look something like this. The nucleus will take up pretty much all of the cell. Okay, so it will be like this. This is the nucleus, and you'll have a tiny little strip of cytoplasm around it. Okay, whereas these germinal center B cells, they'll look something more like this. So they'll have slightly more cytoplasm than the naive B cells, and this is what makes them histologically distinguishable from the naive B cells. And of course, I said that the activated B cell had gone to the periphery, so the germinal center will most likely be around the periphery rather than in the center like that. Okay, so you'll then get this germinal center being formed, and it's full of these germinal center B cells. Now, these germinal center B cells are not all genetically identical. Instead, they are based on the original B cell, but they're not genetically identical to the B cell, the original B cell. What happens is they undergo slight mutations in the antigen binding region of their B cell receptor, okay, and they'll all have slightly different antigen binding domains on the B cell receptor. Okay, so this gives you a B cell population within the germinal center that is all that has a lot of variation within it. All of them have different B cell receptors. Then what you start off is a process of natural selection, basically. Okay, so there is allergen bound to surface proteins on follicular dendritic cells, which are within the B cell follicles. Okay, but there is a limited amount of allergen, and there is a huge number of these germinal center B cells. Now what's going to happen is that they're all going to compete for binding to this allergen, and those that have the best B cell receptors for binding to the allergen, the highest affinity B cell receptors, will bind to the allergen, and those that have lower affinity B cell receptors will not get the allergen. And in order to survive, you need to get the allergen. So those B cells which have the highest affinity B cell receptor for binding to the allergen, those will get the allergen, and they will go through this same process again. They will internalize the whole B cell receptor with the allergen bound to it into a endocytic vesicle. They'll break down the allergen, present it on MHC class 2 along with co-stimulatory molecules, and go and get T cell help, basically, which will trigger them to survive and also to proliferate. Okay, whereas these B cells that have less good B cell receptors, those will not get allergen, and then they will undergo apoptosis. Okay, so they will not survive. So what this gradually favors is a B cell population which have very high affinity B cell receptors. So gradually the population will evolve and evolve and evolve so that the B cells get higher and higher affinity B cell receptors on their surface. And this process where you're gradually making the B cell population B cell receptors higher affinity is what's known as multiple step affinity maturation. And I think there is pretty strong consensus that multiple step affinity maturation does occur in lymph nodes. Now, it was controversial for a very long time. It still is controversial, but I think it's growing. There's a growing consensus that this is what happens. Okay, so multiple step affinity maturation is what this is called. Right, so what happens next? Well, B cells can start to come out of the germinal center, so that in the germinal center, what is gradually happening is this affinity maturation process, which goes round and round in circles, and you're just getting gradually more and more higher and higher affinity B cell receptors in that B cell population. Okay. Now, what will eventually happen is some of these B cells in the germinal center will differentiate into what are known as plasmablasts. So from time to time, what will happen is the B cells, these germinal center B cells, okay, so let's say here we have a germinal center B cell, it can differentiate into a plasmablast, and what triggers it to differentiate into a plasmablast is not fully understood, okay, so, but some of them start to do this. Okay, so here is a plasmablast. Now, plasmablasts stay in the lymph node, and what they can do is they can still divide a little bit, so a little bit of division might occur, so this plasmablast might divide a little bit, but when it divides, it produces cells that are genetically identical to it. So once you've become a plasmablast, the somatic hypermutation stops, basically. Okay, so your B cell receptor is now fixed. Okay, so they're all going to have the same antigen binding region on their B cell receptors. And then you've got a little population of plasmablasts. So now we've got a few more of these identical plasmablasts. And now what can happen is these plasmablasts can then differentiate into plasma cells. Okay, so they're going to differentiate into plasma cells. And I should draw these a slightly different shape. Okay, I should have drawn the plasmablasts a slightly different shape as well. Plasmablasts and plasma cells all have a sort of egg shape rather than an oval shape, a sphere shape. Okay, so like this. An ovoid, I think it's called. The plasmablasts should have that shape as well, never mind. Okay, anyway, these plasmablasts are going to differentiate into these plasma cells here. Okay, right, and these plasma cells will leave the lymph node and go into the blood, and they circulate in the blood for a while, but eventually they take up residence within the bone marrow. So there are places in the bone marrow where they'll take up residence. And what do they do in the blood and the bone marrow? Well, they start secreting antibody. Okay, so let's go over the structure of antibody, because this is quite important. Right, okay, so antibody then. Antibody is basically a secreted version of the B cell receptor. Okay, so it's made by the same gene, and I'll explain this in a moment. Okay, so antibody molecules consist of 4 chains all put together. Okay, so you have 2 what are known as heavy chains. Okay, so let me highlight these in now. So 2 heavy chains. Okay, so here is one heavy chain, here in pink, and it's connected to another heavy chain here by a disulfide bridge that's in between the 2. Okay, so you have 2 of these heavy chains, and these 2 heavy chains are absolutely identical to one another. Okay, then you also have 2 of these light chains, which I'll colour in vivid purple here. And these are connected to each of the heavy chains, so each heavy chain has a light chain which is connected to it again by a disulfide bridge. Okay, now both the heavy chains and the light chains have what are known as variable and constant regions. Okay, so let me draw another picture here. So let me draw the antibody again. Okay, so let's now split up the heavy chains and the light chains into their fixed and variable regions. Okay, so let's first look at one of these heavy chains. So let's pull out one of these heavy chains. Okay, so you can divide it up into 2 portions. This is the variable portion of the heavy chain up here, close to where the antigen is going to bind. So remember, the antigens bind up in these portions here. Okay, these are the 2 antigen binding sites of the antibody. Okay, and this portion down here, after this line here, is the constant region. So you have a variable portion and a constant portion. Okay, the same is true for this light chain here. So here is the light chain, and again the 2 light chains will be genetically, they'll be identical proteins to one another. Okay, and the light chain here, it again has a variable region up here and a constant region. Now, the 2 variable regions, the variable region of the heavy chain and the variable region of the light chain, they come together to make the antigen binding site, which remember is called FAB, the fragment that binds the antigen, fragment antigen binding. So the big F means fragment, and then the subscript AB means antigen binding. Okay, so this is the antigen binding fragment. Okay, and then the constant regions. Well, actually, firstly, just before I talk about the constant regions, I should say you have 2 antigen binding fragments. You have another one over here, again, that's identical to this one over here. So both of those sites are capable of binding to the same antigen. Okay, specifically to the same epitope of the antigen. Okay, which just means that they bind to the exact same site of the antigen. They don't bind at different sites to the antigen. They bind to the exact same epitope, as it's called, of the antigen. Right, now, the constant region then of the light chains and the constant regions of the heavy chains, they come to make what's known as the FC region, which means the fragment crystallizable of this antibody. Okay, so this is the portion that if you get a huge amount of antibody, you can crystallize the FC regions because they will generally be the same, although this is going to be slightly more complicated. We're going to see that there are different fragment crystallizable regions that you can use. Okay, and we'll come to this in a moment when we talk about isotype switching. Okay, so fragment crystallizable. Right, so this entire process of affinity maturation and enzymatic hypermutation, this was occurring with the fragment antigen binding. So you were not altering the constant regions, you were altering the variable regions. So every B cell in the B cell follicles, they will all have different antigen binding regions here, but the constant regions will remain the same. Okay, so that's why they're called the constant regions and these are called the variable regions. Now, it's slightly more complicated than that because there are different what are known as isotypes of antibody. Okay, so let me explain this by talking about the structure of the heavy chain, so the immunoglobulin heavy chain here. Okay, so the immunoglobulin heavy chain, then, if we look at its gene, okay, so let's draw the gene for the immunoglobulin heavy chain. Okay, so let's say this line represents some DNA. Okay, now what happens is that firstly, you have the portion of the DNA that codes for this variable portion of the heavy chain. Okay, so this portion up here is coded by this portion of DNA. Okay, and this is the portion that will vary between every single B cell in the B cell follicles, okay, and it will vary between all the germinal center B cells, etc. Okay, then after this, you have the portion which codes for the constant portion. However, there are absolutely loads of these, so let me discuss this with you because this is quite complicated. Okay, so there is what's known as the constant mu portion first. Okay, then after that, and by the way, these little bits that are in between, those are introns. So you have, within this gene, you have introns and exons. The introns are the portions that are junk DNA, effectively. They're not going to be used to actually make the protein. So the squares that I'm drawing, these represent the exons, and the lines, those represent introns. Then the next thing that you have is what's known as the constant delta portion. Okay, and then it goes on basically. So let me draw them all out and then I'll explain them. Okay, so then the next one is what's known as the constant gamma free region. Okay, then after that you have the constant gamma 1. Okay, then the constant alpha 1 here. Okay, then after that the constant gamma 2. So let me put this here. I'm getting a little worried that it's not all going to fit in now. Constant gamma 2. And don't worry, you don't need to memorize the order of these. You can if you want, but the understanding of what this means is more important, so we'll come to that in a moment. But to understand what this means, I will at least draw it out once. Okay, so then what we have is the constant gamma 4. Okay, and then finally you have constant epsilon over here. Okay, and then finally constant alpha 2 right at the end over here. Okay, right, now what does this mean? What does this mean? Well, basically, all of these code for constant regions of the immunoglobulin heavy chain. Oh, and by the way, immunoglobulin is another word for antibody. So if people are being fancy, they'll say immunoglobulin rather than antibody, but it means this structure here as well. So the immunoglobulin heavy chain locus on the DNA is what this is. Okay, so we have a lot of introns and we have a lot of exons as well. Right, so you have to use this variable region. This is the portion that is specific to our germinal center B cell. So we've picked this one GC B cell, and this is the variable region it's got. Of course, all the other GC B cells will have different variable regions, but then absolutely all of them have these same constant regions behind. So all of these are genetically identical among all of the GC B cells. Okay, now, what happens? Well, you don't need to use all of these to make the constant portion of the heavy chain. You only need to use one of them. Okay, so why do you have 9 of them if you only need to use one of them? Well, you can use the different ones. Okay, so let me explain something. So, when you had a B cell originally, let's say we have our naive B cell here in the lymph node. Okay, so here is our naive B cell in the lymph node. So it has hardly any cytoplasm, a massive great nucleus. Now all of the naive B cells within the lymph node, they have different B cell receptors, okay, and the B cell receptor is made up of these heavy chains and these light chains, okay. Now the light chain is far simpler, it doesn't have all of these different things. So you have one constant chain, you have one constant portion for the light chain, and that's just that, okay. Whereas the heavy chain has all of these differing ones here, okay? So what happens? Well, it's going to have a certain variable region that is specific to this specific naive B cell, okay? And then what's it going to do? It will continue on transcribing this gene. It will go over this exon and then into this constant region here, this constant mu region. And then usually what happens is it stops at transcribing after that, and then the mRNA will go off to the ribosome. It will be translated into a protein, and what will happen is the immunoglobulin heavy chain that you'll produce will have the variable region, and then it will have this constant mu portion here. Now when you use the constant mu portion as your constant portion of your heavy chains like this, you end up with an antibody that is called an immunoglobulin M antibody. Okay, now every single one of these constant regions has a portion right at the end which can be included or not included. Okay, so each one of them at the end has a portion that can either be included or not included, and this is the portion that mounts the antibody onto the cell membrane rather than letting it be soluble. Okay,